Hello and welcome back to everyone. Today we are going to be discussing about immunoprecipitation. This is going to be a brief discussion and it is one of the applications of Western blotting and SDS page. Some of the things that we have learned so far. So delving right into it without wasting much time, immunoprecipitation. So what is immunoprecipitation? Immunoprecipitation or IP is the technique of precipitating a protein antigen out of solution using an antibody that specifically binds to that particular protein. So if you have gone through the previous lecture that I uploaded on Western blotting, you would have heard me mention the term immunoprecipitation and very briefly and in short describe what it actually means. So if we break the word and look into the etymology of the word, we have immuno, which means the use of antibodies, and we have precipitation. So immunoprecipitation refers to precipitating a protein of choice, the antigen, out of solution with the help of antibodies. This process can be therefore used to isolate and concentrate a particular protein from a sample containing many thousands of different proteins. So how can you isolate? So if you have a mixture of say thousands of different proteins, your antibody, due to its characteristic nature, will bind specifically only to its antigen. And now when you precipitate the antibody out, the antigen will also be precipitated out along with it. So this will help you to isolate a specific protein. Now coming to concentration. So say for instance, say you have a 1 ml solution and you do immunoprecipitation, you precipitate the protein out. Now once you have the precipitate, you resuspend that in a much smaller volume, say 30 or 40 microliters. So now your protein has gotten concentrated from 1 ml solution to 40 microliters. So say you had X amount of protein in 1 ml and now you have 40 the same amount of protein in 40 microliters. So it has gotten concentrated several folds. Now, IP can also be defined as the small-scale affinity purification of antigens using a specific antibody that is immobilized to a solid support. Now, why do we need the solid support? We'll discuss about it. IP requires that the antibody be coupled to a solid substrate at some point in the procedure. Why? Because if you think about it, the antibody molecules with which you're fishing or pulling out the protein out of solution is also a protein molecule by itself. So it will remain in solution unless and until you couple it to some solid substrate. Now because it is coupled to a solid substrate, this solid substrate will settle down under the effect of gravity when let by itself, or you can settle it down by simply spinning the tubes, centrifuging the tubes, or by using specific stands which are magnetic stands in case your solid substrate is magnetic in nature. And as this substrate settles down, the antibody also settles down along with it, and so does the bound antigen. Typically, the antibody is coupled to protein A or protein G sapphirose beads. Antibody conjugated to agarose beads or magnetic beads can also be used. But for using these magnetic bead conjugated antibodies, you will be requiring specific magnetic stands. IP is one of the most widely used methods for isolation of proteins and other biomolecules from cell or tissue lysate for the purpose of subsequent detection by Western blotting and other assay techniques. So if we take a look at a schematic representation of the IP protocol, so first you begin with your cell lysate, which has your total protein, that is every protein present in the cell. You have lysed the cell, you have released the intracellular contents, and these are now in solution. So you have a solution of different proteins and you have this red one, which is your protein of interest. So you add antibodies and you incubate along with the antibodies. So this incubation will allow an antigen-antibody complex to form, wherein the antibody will go and specifically bind to your protein of interest. In the next step, you add protein A or protein G beads and you incubate again. Well, now what will happen? This protein A or protein J beads, they have a characteristic feature of binding to the Fc portion of antibodies. So they will not specifically bind to Fc portion of any antibody. So it is bound now. Now you wash and elute So in washing step, what you do is that you remove these excess unbound proteins or anything that was non-specifically just sticking to your antibody during washing steps, that would get removed. And then you elute your protein. Why do you elute? Elution is required to separate your protein of interest from these antibodies which are bound to the beads. And once you have your purified protein, you can simply run it on a gel, do a Western blot, or you can simply visualize it directly by Kumasi staining. So next, coming to the types of immunoprecipitation. Immunoprecipitation can be of the following types: individual protein IP, co-immunoprecipitation or co-IP, chromatin immunoprecipitation or chip, and RNP immunoprecipitation or RIP. So to begin with, we will first discuss about individual protein immunoprecipitation. Now this involves using an antibody that is specific for a known protein to simply isolate that particular protein out of a solution containing many different proteins. So the schematic that we looked at just now, or the methodology that we discussed just now, that is the underlying principle for simple individual protein immunoprecipitation, wherein you know you have a particular protein in solution and you want to isolate that protein from that solution, so you add antibodies specific to that known protein. These solutions will often be in the form of a crude lysate of, say, a plant or animal tissue. Other sample types could be body fluids or other samples of biological origin. Next, we will move on to co-immunoprecipitation or co-IP. Now, immunoprecipitation of intact protein complexes, that is, antigen along with any other protein or ligand that is bound to it, is known as co-immunoprecipitation. So the co-IP method is commonly used routinely in many laboratories to study the interaction of two or more proteins. So this is something that I already discussed with you in the previous presentation on Western blotting very briefly when talking about fluorescence based detection and the advantage of being able to detect multiple bands simultaneously in a blot. So in co-immunoprecipitation, say you have two proteins, protein A and protein B, and or rather, let's go in this way. Let's say you have a protein A and you know that it is interacting with certain proteins, but you do not know what other proteins it is interacting with. In that case, you can do a co-immunoprecipitation experiment. You simply pull protein A out of solution using antibodies specific to it, and along with it, any interacting partner will also be pulled out. So co-IP works by selecting an antibody that targets a known protein that is believed to be a member of a larger complex of proteins. By targeting this known member with an antibody, it may become possible to pull the entire protein complex out of solution and thereby identify unknown members of the complex. This works when the proteins involved in the complex bind to each other tightly, because there are several steps in the IP protocol like we just discussed. There is, after binding, you allow for several steps of washing to remove anything that is bound non-specifically, and then you elute. So there are multiple steps involved, and in order for you to be able to detect the binding partners of this protein A, you require that this interaction be strong enough to survive the various steps of the protocol. So only when they bind to each other tightly, it makes it possible to pull multiple members of the complex out of solution by latching onto one member with an antibody. This concept of pulling protein complexes out of solution is sometimes also referred to as a pull down. So an IP experiment or an IP assay can also be referred to as a pull down assay, but this is a very colloquial term and it might be used internally in laboratories but never in a formal setting. So co-IP is a powerful technique that is regularly used by molecular biologists to analyze protein-protein interactions, and I for a fact can say that this is one experiment that I do very routinely in the lab owing to the kind of work that I'm currently engaged in. So if we take a look at the schematic of co-IP, so it is the overall layout is similar to the one that we observe for individual protein immunoprecipitation. But here, in addition to the red protein of interest, which was say our protein A against which we are adding the antibodies, we have another protein, this yellow protein, let's call it protein B, which is also binding to protein A. Now, I don't know if under a given experimental condition, if this protein A and protein B interaction is taking place. Say for instance, the aim of my experiment is to identify which residues are critical for this protein A protein B interaction. So how do I do that? One easy way could be a co-IP experiment. How so? So one by one, I say for instance, suspect that there are 10 residues which can be involved in this interaction or which can be important for this interaction. So now I will go about mutating these 10 residues to see what kind of effect this mutation will have on the binding interaction. Now, so this now becomes my test condition. So I have a range of protein A mutants, and now I want to see which of these mutants are still capable of binding to protein B and which have lost this binding ability. So under such an experimental situation, co-IP experiment becomes very useful. So those mutations which do not interfere with the binding interface will not disrupt this complex formation, and those mutations which do interfere with it will disrupt this complex formation. So next you add antibodies specific for this red protein, which is your protein A, and you incubate with it. You add this protein A or G saccharose beads and you incubate along with it as well. You wash to remove any excess proteins that are freely moving around in the solution or anything that is sticking non-specifically to your beads. And over here I would like to add that typically in your washing buffer, keeping keeping a little amount of detergent, say for instance 0.01% or 0.001% even, can be useful because detergents help in ensuring that anything that is binding non-specifically will get washed out. And finally, you elute and purify your protein of interest. You run a gel to visualize it. And if you want, you can do a Western blot as well. But do keep in mind that in order to be able to do a Western blot, you should be already aware of the identity of this interacting partner, protein B. If you don't know what proteins your protein of interest is interacting with, you can't really visualize it with a Western blot because you can't use antibodies against it if you don't know what the antigens are. But if you do know that, you can go on to do a Western blot. So the third kind of immunoprecipitation experiment that we are going to talk about is chromatin immunoprecipitation or CHIP. So chromatin immunoprecipitation or CHIP is a method that is used to determine the location of DNA binding sites on the genome for a particular protein of interest. So right now, up until now, we were looking into protein-protein interaction, but CHIP is a method which is used to study protein-DNA interaction. And by this method, you can identify the DNA binding sites for a particular protein of interest. If you have a known protein which you know binds to the DNA, you can identify where exactly it is actually binding. So this technique gives a picture of the protein-DNA interactions that occur inside the nucleus of living cells or tissues. The principle underpinning this assay is that DNA binding proteins, including your transcription factors and histones in living cells, can be cross-linked to the DNA that they are binding. Typically, formaldehyde is used for this purpose. Now, it is very crucial to keep in mind that if you do not do this cross-linking, then this interaction is not tight enough to survive the different steps of the IP protocol. So this cross-linking is very, very important. And this cross-linking makes it possible to do such interaction studies. Also, over here, I would like to add something that I missed out, that when studying protein-protein interactions by co-immunoprecipitations, sometimes some interactions might be very transient or they might be not so stable, not so tight. So you can stabilize that interaction by cross-linking the proteins as well. You can use several cross-linkers which simply non-specifically cross-link the protein based on their proximity, and then you can perform the IP experiment. And several of these cross-linkers also contain these reducible disulfide bridges. So when you treat the samples to prepare them for loading onto the SDS page. Your beta mercaptan ethanol present in the Lamley's buffer will reduce this disulfide bridge, and it will separate out these chemically cross-linked interacting proteins, so that now when you visualize them on either the gel or the Western blot, you can identify the protein on the basis of its size. You will not be observing a full complex, but you can get the individual proteins involved in the interaction on the gel. So by using an antibody that is specific to a putative DNA binding protein, the protein DNA complex is immunoprecipitated out of the cellular lysates. Following cross-linking, the cells are lysed and the DNA is then broken down into pieces typically 0.2 to 1 kb in length by sonication. Now, why do you have to break down the DNA? Because as you know, genomic DNA is like really long, and if you want to identify just a specific portion of the genomic DNA that is interacting with a protein of interest, you do need to shear it. Otherwise, you will just end up pulling down the entire genomic DNA. The purified protein DNA complexes are then heated to reverse the formaldehyde cross-linking. Now this is very important. Whatever cross-linking you are doing, you should be able to reverse it. Otherwise, you cannot identify this DNA fragment that is co-precipitated along with your protein of interest. So you heat the purified DNA protein complex to reverse the formaldehyde cross-linking, which allows the DNA to now be separated from the proteins. The identity and quantity of the DNA fragments isolated can then be determined by simply doing a PCR. But yes, keep in mind, in order to be able to do a PCR, you should also have some basic idea about what DNA fragment your protein of interest is binding to. Because unless and until you know that, you cannot design your primers for doing the PCR. So this you have to keep in mind. So this is kind of similar to knowing what the interacting protein is in case of co-IP if you want to do a Western blot. The limitation of performing PCR on the isolated fragments is that one must have an idea which genomic region is being targeted, like I just mentioned. And sometimes this limitation can be circumvented. How? You simply clone the isolated genomic DNA into a plasmid vector and then using primers that is specific to the cloning region of that vector. So yes, it is quite natural if you are doing some experiment for the first time and say for instance you have a novel protein. You know it binds to the genomic DNA, but obviously you have no idea where it binds. Then how will you go about it? You want to identify this DNA fragment where it binds. So in order to allow that, you can, after you have isolated the DNA fragment, you can clone this DNA fragment into a plasmid vector. And now because you know what cloning sites you have used, you know the sequence of the vector, you can design primers that will bind to the vector near the cloning sites. And now you can simply PCR amplify this cloned genomic DNA fragment. PCR amplification by itself is not enough for knowing the sequence of the genomic DNA. Once you PCR amplify, you then once again clone it and you send it for sequencing. And once you sequence the DNA, that is when you have the final identity. So if we take a look at the schematic representation of chip, so you have your cells. Before you lyse the cells, the first and foremost step is to cross-link by treating with formaldehyde. In the major advantage of this kind of cross-linking is that no prior modification of either the protein or the DNA is required. You simply add formaldehyde and it will cross-link the genomic DNA with whatever proteins are in close proximity to it. Then you lyse it. Once you have lysed the cell, you fragment it. It is not really mentioned here, but you have to shear the genomic DNA. Then you allow your antibody to bind to the protein of interest. You do your immunoprecipitation where you see along with the antibody, the protein bound to it and the genomic DNA bound to it have settled down, and you have the rest of the sheared genomic DNA fragments floating. You wash to get rid of this, and then you reverse the cross-link by heating. You have now separated your protein and your DNA fragment. So for the protein, you can simply do a Western blot or do a coumacy gel to confirm that yes, indeed, your IP protocol was successful and you have successfully isolated the protein out of solution. And then you purify the DNA and run a quantitative PCR. So the next kind of immunoprecipitation that we are going to talk about is RNP immunoprecipitation, or also abbreviated as RIP. Now, RNP immunoprecipitation targets ribonucleoproteins or RNPs. Live cells are first lysed, and then the target protein and associated RNA are immunoprecipitated using an antibody targeting the protein of interest. So this is in a way very similar to CHIP. You can say this is a modified version of CHIP, wherein in CHIP you were studying the interaction between chromatin and a protein, and in case of RIP you study the interaction between a target protein and associated RNA. The purified RNA protein complexes can be then separated by performing an RNA extraction, and the identity of the RNA can be determined by cDNA sequencing or RT-PCR. So if you take a look at the schematic that is shown over here, so you have your cells. You can either go for it directly, where you simply lyse the cells, do the RNA immunoprecipitation, you decross-link. Yes, cross-linking is also very critical in case of RIP, just like in CHIP, and you have the purified RNA. Or you can do this nuclear cross-linking, where you basically isolate the nuclei and begin with there. So in the native kind, you are simply isolating the entire cell lysate, that is both the nuclear content as well as the cytoplasmic content. But in this case, you're isolating only the nucleus and cross-linking it. And then you shear the chromatin and digest the DNA. You do RNA immunoprecipitation and you decross-link and you have your purified RNA. Why is this chromatin shearing and DNA digestion necessary? Well, chromatin shearing is required to allow for efficient DNA digestion, and DNA digestion is done simply to chop off the DNA into much smaller fragments so that they do not interfere with this process of isolating this RNA bound to its target protein. This DNA, once it is digested and sheared, can be easily removed out of solution. So the difference between the two, or what would be one advantage of one over the other, is that in case of nuclear cross-linking, you only get to identify protein RNA interactions that are occurring in the nucleus. But as you know that RNA, it doesn't only reside in the nucleus, you have RNA in the cytoplasm as well. Messenger RNA is always going to the cytoplasm. So if you want to find out the interaction between any RNA and protein occurring in the cytoplasm, in that case, you go for this native RIP. And then once you have the purified RNA, you can do either real-time PCR, Q is quantitative real-time PCR, or you can do a microarray or high-throughput sequencing to identify this RNA that you have over here. So next, you have a modified version of RIP, which is known as PAR-CLIP, or photoactivable ribonucleoside enhanced cross-linking and immunoprecipitation. So this is very up and coming and is increasing in popularity these days. So the method relies on the incorporation of photoreactive ribonucleoside analogs such as 4-thiouridine 4-SU and 6-thioguanosine 6-SG into nascent RNA transcripts by living cell. So in the first step of doing PAR-CLIP is that you have to incorporate these photoactivable ribonucleoside analogs into your nascent RNA transcripts. So basically you have to grow your cells in the presence of these analogs, so that when the cell is growing and when the cell is replicating and dividing and carrying on its many processes, and in that process it is synthesizing RNA, these ribonucleoside analogs will get incorporated within the RNA being synthesized. Irradiation of the cells by ultraviolet light of 365 nanometer wavelength induces efficient cross-linking of these photoreactive nucleosides, labeled cellular RNAs, to interacting RBPs. So once you have these analogs incorporated into your RNA, so when you irradiate such cells with UV light, what happens is that these photoactivable ribonucleoside analogs get activated, and whatever protein is present in its close proximity, it will simply cross-link with those proteins via interaction with specific amino acids and specific groups, to be more precise, of the amino acids. And now you have your RNA and your interacting protein cross-linked to each other. So if we take a look at the schematic shown over here, you have these HEK239 cells. So HEK239 are human embryonic kidney cells, which are commonly used in many laboratories. So you have your HEK cells, which you culture in presence of 4SU, which is this photoreactive ribonucleoside analog. So now when you culture it, whatever RNA is getting synthesized, it will utilize this analog present in the media and incorporate it into the RNA. Next, you irradiate it with UV light of 365 nanometers, and this will lead to cross-linking of these modified RNAs with the interacting protein partner. So you see, this is the protein which is shown as a gray blob over here, and you have your RNA cross-linked to it. So you cross-link it, you lyse the cells, you do your immunoprecipitation, and you also do an RNase treatment. Now, why is this RNase treatment essential? Because you only want to identify the specific region of the RNA where your protein of interest is bound. So doing this RNase treatment becomes critical to get rid of the excess overhanging RNA, and also any other RNA that would be present in solution. You can get rid of that by subjecting it to RNase treatment. Next, in this particular protocol, what they have done is they have phosphorylated the 5' end of the RNA so that now they radiolabel it. So first they phosphorylate the 5' end and they radiolabel it while doing that because they use radioactive P32 phosphate for phosphorylation. So next they run an SDS page to separate out the protein of interest, which is bound to the RNA, and this protein is something which you're already aware of. You know what protein it is because you're using antibodies against it. Then you do a proteinase K treatment. So once you've run it on the gel, you still have the complex on the gel because you did not decross-link it, mind you. So whatever you get on gel, that is the protein RNA complex. So next, what they did was they isolated this protein RNA complex from the gel, and now they subjected to proteinase K treatment to simply digest and remove the bound protein, and then they recover the RNA. And once they have recovered the RNA, they simply, in this case, ligate and adapt it to it. This adapter ligation will allow for subsequent cloning and then subsequent sequencing of the RNA that has been isolated. So another kind that we are going to talk about is tagged protein immunoprecipitation. So what is tagged protein immunoprecipitation? One of the major technical hurdles with immunoprecipitation is the great difficulty in generating an antibody that specifically targets a single known protein. So whatever we have discussed so far was dependent on having an antibody that was binding to the specific protein, and then you were identifying other interacting partners of that protein, whether those partners be proteins or DNA or RNA. But you have to keep in mind that it is actually difficult to generate an antibody specific for each and every protein that you want to study. And also it's not very efficient, neither in terms of cost nor in terms of the amount of time and energy that it would take, the amount of resources that this demands. So to get around this obstacle, tags can be engineered onto either the C or the N-terminal end of the protein of interest. So the advantage of this method is that the same tag can be used again and again on many different proteins. So if you can add a tag to your protein, which you want to then isolate out of solution and whose interacting partners you want to identify, and you have antibodies against that tag. So this tag now becomes common, and no matter what be the number of proteins you want to study, you can simply add this tag to each and every one of these proteins. So now you have these common antibodies that you can use for all of these experiments. So instead of requiring 10 different kinds of antibodies, one antibody does the job. So examples of tags that are commonly used for such IPs are GFP tag, the green fluorescent protein, GST tag, glutathione S transferase, and the flag tag. However, it is very important to keep in mind that the use of a tag to do pull down assays actually is very common, and the use of a tag makes it very convenient. it does raise some concerns regarding the biological relevance because the tag itself may either obscure native interactions or introduce new and unnatural interactions. So you always have to keep in mind, and this is the same for introducing tags to proteins in case of any experiment you want to perform, regardless of whether it is IP. For any experiment you want to perform, the moment you introduce a tag to your protein, you have to keep in mind a couple of things. First, will the tag interfere with proper folding of your protein? Second, will your protein be functional despite the presence of the tag? Third, if the protein is supposed to interact with another protein, will the tag somehow interfere with this interaction? Will it obscure it or will it promote it? So these points one has to keep in mind. So if you take a look at the schematic of it, so you in this case have say S tag, right? And you have your bait protein. So it doesn't matter what the S tag is, let us just call it S. And you have your bait protein. So yes, something I forgot to mention previously, that in immunoprecipitation experiments or co-immunoprecipitation experiments, the protein which you're pulling, that is the protein against which you have the antibody, is called the bait. And with this bait you capture other interacting partners. So you simply incubate this S-tagged bait protein along with anti-S-tag coated beads. The purpose here is to immobilize the protein for binding. Next you add your prey protein which you want to capture, and this prey protein has a flag tag. So now you have this anti-S-tag coated bead on which this bait protein which was conjugated to S-tag is immobilized, and you add your prey protein. Next, what you do is that you perform washes to reduce any non-specific binding, and you elute the bound proteins from these magnetic beads. Okay, so in this case, these beads were magnetic beads. So you elute these proteins from these magnetic beads, or regardless of what the nature of the beads are, let us go with that you elute them from the beads. So now you have your entire complex separate from the bead. And now you can simply analyze the binding through a Western blot using tag-specific antibodies. So in this case, because your prey protein has a flag tag, so you can use anti-flag antibodies. So this is also very beneficial where your prey protein has a tag, because then now you can use one common antibody for several different prey proteins which are tagged with the same common tag. So one more thing that I would like to cover in this lecture is direct versus indirect IP. So the two general methods for immunoprecipitation are the direct capture method and the indirect capture method. So what are these? Coming to the direct capture method. So antibodies that are specific for a particular protein or group of proteins are immobilized on a solid phase substrate such as superparamagnetic beads or microscopic agarose beads which are non-magnetic. The beads with the bound antibodies are then added to the protein mixture, and the proteins that are targeted by the antibodies are captured onto the beads via the antibodies. In case of indirect IP, you first mix the antibodies with your protein. So antibodies that are specific for a particular protein or a group of proteins are added directly to the mixture of protein. And once this antigen-antibody interaction is allowed to complete, the beads are then added. So the antibodies are not attached to a solid phase support at this point. They are free to float around the protein mixture and interact with and bind to their targets. Beads coated with protein A or G are then added to the mixture of antibody and protein. So the schematic will help make it much more clear. In case of direct IP, you add antibodies that are already conjugated to the beads. And in case of indirect IP, you first allow the antibody to interact with the protein of interest in solution, and then you add the beads. So if you think about all the schematics that we covered so far in this lecture in the earlier slides, those were all examples of indirect IP, where you first allowed the antigen-antibody interaction to take place and then you added the beads. Whereas in case of direct IP, you add antibody already conjugated to the beads, and then the rest of the steps are same. So you have your antibody conjugated to the bead, you allow it to bind to the protein of interest, and you elute it to have the purified complex. or you can simply add the antibody first and let it interact with the antigen of interest, and then you add these specific beads which will bind to the antibody, and you purify the complex. So this was our discussion on immunoprecipitation. I tried to touch upon the basic principles and also the common different types of common different modifications of IPs that are done. In case you have any questions, feel free to leave them in the comment section and I will surely get back to you. And if you like this lecture, do like, share and subscribe for more such content. Thank you, and I'll see you next time.